Hello there, everybody. Cameron Geller here, one of the Bio 107 lab TAs. So I hope that you're all doing well with this new virtual learning. So in addition to some of the other videos we're going to be putting together, myself and the other lab TAs, this is one of the videos you may already have available on Canvas. If not, please speak to your lab instructor to get that video available. Otherwise, I will have it here as well. So for this week's lab, we're not going to be doing too many experiments. It's mostly just going to be an introduction into plant anatomy and physiology. And this is the third and final section of the Bio 107 lab. So with that, let's go ahead and get started with some basic anatomy. So throughout this presentation, I'm going to be showing you pictures of different plants or flowers, the label of which is going to be at the very top here, where you can see the mouse cursor, where in this case it says a Coleus stem tip. And the LS just means it's a longitudinal section. So you're seeing, it's kind of like if you took your arm and you were to cut it down the middle so that you get a longitudinal view or a vertical view of what the plant looks like, as opposed to a cross section where you'll see circular images. So shown here, we have a few key regions I want to identify, starting first with the apical meristem, where the arrow is pointing. So this is the tissue at the top of what's called the shoot of the plant. So plants can be broken up into shoots and roots, where the shoot is the part of the plant above the soil and the root is the part below the soil. So this is the part where stem elongation is going to take place. And so there's going to be a plant hormone called auxin that's going to be secreted by the plant at the apical meristem that allows for the vertical growth of the plant. In addition, down here on the lower right, we have the leaf bud primordium. This is a group of cells that are produced by the apical meristem that will eventually develop into the leaves of the plant. And down here we also see an axillary bud. And this is going to grow from the leaf's axil down here, which may develop into branch or flowers. So we can see from this primordium that up here we have the development of a young leaf that's going to contribute to absorbing light for photosynthesis. And in different plant, we might see the formation of branches or flowers occurring, which we can actually see here on the sides as well. So here's that same Coleus stem tip from the previous slide. This time we've actually zoomed in. So this large hump hill-like structure, this is a close-up magnification of that shoot apical meristem. Again, that's where auxin is produced to allow for the plant to grow vertically as opposed to horizontally, which if it's horizontal growth, it tends to have more of a bushy-like appearance. And in next week's lab, we're going to actually see in detail what that's like. So next, we have a different picture of the Coleus stem tip, this time showing what are called the trichomes. So the trichomes you can think of as being these small hair-like protrusions from the epidermal layer, the outermost layer of the plant cells, that grow out in order to aid in water and nutrient absorption for the plants. So it allows for the plant to communicate with its extracellular environment in order to acquire nutrients needed to survive. So shown here, we have a helianthus stem. This is a sunflower. This is the scientific name for sunflower. And here we have the different layers that break down the plant in further detail. This is a cross section this time, so that's what CS stands for. So now we're seeing a circular view as opposed to a longitudinal view of the plant. So first I'm going to start with the epidermis. So this is the outermost layer of the plant that's going to provide as a key layer of protection against any harmful infections or diseases from killing the plant or making it sick, more or less, kind of like we have the outermost epidermal layer of our skin that protects us. So it's the same function of plants. Next, we have this region inner to that known as the cortex, and this is going to store important molecules that maintain the plant function, which includes storing carbohydrates or sugars, as well as essential oils that help to maintain the overall plant functionality. Over here in this giant circle, we have what's called the vascular bundle. So this is a region that's going to aid in transporting critical substances to various parts of the plants. Kind of like our body has a circulatory system for delivering blood, oxygen, and other nutrients, the vascular bundle is going to carry out a similar function. And this is actually we're going to look at this in further detail on the next slide and what's contained within this bundle. Finally, we have the pith, which is the center, which contains what are called parenchyma cells, and these are cells that help the plant to participate in photosynthesis. So we'll go over that in a little bit detail in just a moment. So now we're zooming in on that region That's the vascular bundle that contains some very important substances that you probably may have recalled learning about in your high school biology class. First, we're going to start with the bundle cap. So this is a layer of supporting tissue within the plant that helps make it stretchable and erect without succumbing to gravity. So in order for the plant to acquire certain nutrients, it needs to be able to stand upright to access materials that could otherwise not if it was lying flat on the ground, and the bundle cap will ensure the integrity of the plant structure for that reason. Next, we have what's called the cambium, this region right here where the mouse cursor is. And this is tissue from which the xylem and phloem cells are going to derive. And xylem and phloem are probably the terms, if any, you're most familiar with in botany. So xylem is a section of vascular tissue in plants that transports water and nutrients from the root. So just like humans, plants also need nutrients that include water in order to maintain vitality. So the xylem cells are going to ensure that that gets transported to wherever the plant needs it the most. Next, we have the phloem tissue. This is going to aid in transporting other important nutrients that include sugars, which are carbohydrates, as well as proteins and mRNA compounds. So you've learned in your biology classes that those are important compounds to keep cells alive, and the phloem is going to ensure that those materials are transported to where they need to get to in any given period of time, depending on the plant's needs. So next we have a Zea mays mature stem. So this is actually corn. And this is again a cross section outlining the different regions. So again, we have our vascular bundle. So we're going to zoom in on this in order to get some more details on what's going on there. One thing I do want to note is that in comparison to the previous image, this one you see the vascular bundle has a very different appearance. And that's because these are what we call monocot plants. So monocots are a type of plant that can be distinguished from dicots. So the main difference for the scope of our class is that monocots organize their vascular bundle differently from dicots. So the mnemonic I was taught when I learned this as an undergrad is that monocot vascular bundles have these monkey faces. So here you can see the eyes, the forehead, and the mouth, and that's how I remembered monkey face monocots in order to determine what you're looking at. And it's important to know that because hint, hint, when it comes to the exam, you'll want to be able to know how to identify a monocot versus a dicot. So if that helps you to remember it, don't forget it. So just like in the previous image, we also have a cortex, which again is going to store important molecules like your oils and carbohydrates. We have our outer epidermal protective layer of cells. And again, we have a pith. And you notice this time around that the vascular bundles are more scattered all over the place, whereas in the previous image, they were a little more organized in our dicot plants. So here's us zooming in on that region from before, the vascular bundle. So again, we see our monkey face in further detail. We have our bundle sheath just like before, which is going to help with the plant's integrity to allow it to stand up erect. Then we have our phloem and xylem cells, which again are going to aid in transporting important molecules. For xylem, that's going to be your water and other important minerals and nutrients, whereas for phloem, that's going to be your mRNA, proteins, and carbohydrates. And then we have this bundle sheath, and bundle sheath is a layer of cells that forms a protective layer around the vascular bundle. So as you can imagine, because the xylem and the phloem and the bundle sheath play critical functions in the plant's ability to survive, it helps that the plants have adapted this layer that serves as a barricade to protect these important tissue layers to ensure that the plant doesn't lose its critical functions. Okay, next we have a longitudinal section of a cucurbit stem. So again, we've already looked at parenchyma cells. These are the cells that are going to participate in photosynthesis. We've also got some what are called sieve plates of phloem. So these are going to be sections right here where the mouse cursor is pointing that is going to allow for the phloem material to be passed more effectively from one piece of phloem tissue to the next. So this is critical for ensuring that material doesn't get lost when it's being transported to different parts of the cell, or excuse me, the plant. And then in addition to a sieve plate, we also have what's called a sieve tube. So this is just a tube structure that, again, just adds to the overall ability for material to be transported effectively in the phloem tissue cells throughout the entirety of the cell and plant body. A new term that we haven't come across yet is called a companion cell. So companion cells, indicated right here, are specialized parenchyma cells that help regulate the flow of nutrients through the sieve tube and the plate of the phloem. So again, going back to the fact that material passed through the phloem is critical to the plant's survival, there's a lot of other surrounding cells and tissue layers that ensure that this process isn't hindered or interrupted in any way. And companion cells are companions that help to carry out this phenomenon. So next we have another longitudinal section of the cucurbit stem. This time we're focusing on, they kind of look like little stitches, like if you sewed up a wound in a hospital, and these stitches are what we call helical elements of tracheid cells in the xylem. So this time we're focusing on the xylem instead of the phloem. So helical elements are going to help to hold the tracheid cells together. So again, just like a stitching for a wound closure, these helical elements are stitching together the tracheid cells. And the tracheid cells aid in the transport of water and mineral salts. So the tracheid cells, again, are contained within the xylem. So while the xylem itself is responsible for transporting water and mineral salts, the xylem is a tissue of cells. And within those cells, we have within the xylem tissue, we have these tracheid cells that specifically carry out this function. Next, we have a tilia, a 3-year stem. And this is a cross section. You can tell because it's circular, so circular cross section, long longitudinal. So again, just some more mnemonics to help you to memorize the terminology. So on this outermost layer, we have an epiderm, just like we did before, and immediately underneath that epiderm, we have a secondary protective layer called the periderm. So peri means underneath. So that's how you can remember that this is the dermal layer of protection underneath the epidermis. Epi means above. So again, prefixes definitely help a lot in your science classes. And again, this is a secondary tissue that serves as a protective layer in addition to the epidermis. Now, this is actually a woody plant. So you can think of it as like a tree, right? So we call these woody plants because it's material that's composed of wood. So that's why this picture actually looks different from all the other pictures. So underneath or peripheral to the periderm is the phloem. So again, the phloem is going to be the area that's transporting the important molecules like mRNA, proteins, and carbohydrates. And then we also have the xylem. So we've already talked about that. What's unique to this woody plant that's different from all of our other plants that we've seen in the previous slides is that we have what are called growth rings. So perhaps if you were a Boy Scout or a Girl Scout, or just going back to like an outdoor science camp, or just as a kid when you were first introduced to science, your teachers may have taught you that you can determine how old a tree is because if you cut it open, you can count these growth rings. And it was understood that each ring represents a year on the tree. So if you've ever gone hiking up in Northern California and you've seen the large 100-footer, sometimes 200 or 300-footer redwood trees, you can imagine how many rings would be there if you were to chop it down. But of course you can't because it's protected as a national landmark. But you could imagine how many rings are there. So if you look closely, you see that the thickness of the rings, as you can see from my mouse cursor, some are thicker than others. And this thickness actually tells you how much water was absorbed by a plant in a given year and how much the plant grew. So for instance, in between these rings, you see how the regions are a little bit thicker, right? So while the number of rings tell you the years of the age of the plant, the thickness tells you how much water absorbed. So the thicker the region in between these growth rings, the more water that was absorbed in that given year, which is why you had more cell growth. Whereas the thinner the regions between those growth rings, it may have been a drought or it was off season, not as much water was available and accessible to the plants. So here's a zoomed in region again of the same image from before. We have our epidermal region layer, our peridermal layer, then we have our phloem and xylem, and then the growth rings. So it's just a zoomed in layer to give you a better idea. Now, similar to before on our non-woody plants, we saw that there was this vascular cambium that divided the xylem and phloem. And so that's exactly what's going to happen in our woody plants here. So again, the vascular cambium, just like in our non-woody plants, is located between the xylem and the phloem, and it's also the source of secondary xylem growth. So again, just like in human cells, you'll have some cells that die and they need to be replaced and replenished. That's also going to happen here via the vascular cambium, where it's going to enable secondary xylem growth. Again, xylem is going to be down here, whereas phloem is going to be up here. So secondary xylem growth is occurring inwards towards the pith or the center of the woody plant. And it's also, the vascular cambium will also facilitate secondary phloem growth, which is going to move outwards towards the epidermal region, towards the epidermal layer, or towards the bark or the woody part of the plant. We also have here what's called a phloem ray. So these are cells that are an extension of the phloem cells that are going to radiate outward from the center. So that's going to be the new phloem cells that are being produced under cell division. So just like a phloem ray, we also have a xylem ray shown here, which just like with the phloem rays, these are going to be extensions of xylem cells that radiate towards the pith of the plant. So that's the main difference in woody plants between the xylem and the phloem rays. Phloem rays are going to radiate outward, whereas xylem ray cells are going to radiate inward. Why that is simply because the cells have different functions in transporting different molecules that require them to differentiate and relocate to different parts of the plant. Okay, so now we're going back to our non-woody plants. Here we have a mature buttercup root, ranunculus, and this is a cross section. That's why you can tell because it's a circle. And what you may notice this time around that's very different from our monocot plants, this is a dicot, I should mention, is that in this case, we don't have our xylem and phloem region or our vascular cylinder spread out throughout the entirety of the plant, nor do we have it giving this monkey-like appearance. In this case, we actually have quite an abundance of parenchyma cells. And we just have this tiny little vascular region in the center, and then of course our epidermal layer. Why we have this difference, again, different plants have different functions and live in different environments, and so they'll change their physiology in order to acclimate to their environment. And that's an important concept in plant and animal physiology, which is cell plasticity. It's the ability for plants and animals to respond to their environment and change their morphology. And one of the other videos we're going to post on Canvas will go over that in further detail. So let's go ahead and actually zoom in on this cross-sectional in a little bit more detail. So in this case, we have all of our parenchyma cells. And as I said, they do participate in photosynthesis. And what you're seeing here, if we zoom in closely, are starch granules. Starch is sugar, right? And it makes sense that we should see a lot of sugar here because photosynthesis produces sugar. And so the staining in purple identifies all the sugar that's present, so that's how we can identify that these are parenchyma cells. Now, unlike our previous plants, which had a smaller cortex region, this cortex region in this buttercup ranunculus root is much, much larger. Now, what's interesting here is the fact that we have so many parenchyma cells, it must mean that this part of the plant, or this plant in general, must do a lot of photosynthesis. Why that is, maybe they require more photosynthesis because they need more energy than other plants around them. Maybe they're less efficient at using their energy. It's an educated guess that basically just goes to the idea that plants are acclimating to their environment by changing the behaviors that they perform. So here's a zoom in on that vascular cylinder in the center of our buttercup root. So, excuse me. So unlike our woody plants where the epidermal layer started way out in the periphery of the plants, here they don't start until the very center. So it's a different organization, but the same functions. So we have our endodermis, which is going to be right underneath our epidermis and in between our peridermis. And then underneath the endodermis, we have what's called the pericycle. So this is going to provide support, structure, and protection for the plant to allow for it to stand upright. So it has a similar function to the bundle sheath that we saw from before. And then this time we have the xylem and phloem cells, and they're organized differently, right? Because this is a dicot, so we're not seeing that monkey face. The way that I showed students how to memorize this that I learned as an undergrad is that the xylem are these big circular like window cells, and if you notice, they form a plus sign, or if you rotate it a little bit, it looks like an X. So X for xylem, so that's how I memorize these cells. And then the surrounding cells outside of that X are your phloem cells. It's a little cut off here on the PowerPoint, but you can see most of the spelling. So next we have a zea root. This is a cross-sectional once again. And as you can notice, this time we have a vastly different organization where in this plant, it's still a dicot because we're not seeing those monkey face formations, but we do see a different organization of the vascular cylinder, which contains the xylem and phloem. But again, it's still a dicot, but in this case, this particular plant just has more vascular cylinders, whereas the previous image had only one. So again, just plants adapting respectively. We also have our pith, again, the center where our parenchyma cells are located, this cortex region, and then the outer epidermal layer of protection. So it's a little cut off in this slide, but this is the same picture from before. Now we're just identifying the xylem, which again are the larger window openings, so to speak, and then the phloem is surrounding that. And then we have our endodermis and pericycle, just like in our previous dicot plant. It's just, again, it's distributed differently, but the functions are all the same. Okay, so next here we have a cross-section of a privet leaf. So this is just basically a leaf that was cut from the side so that you can see what the different layers, cell layers look like on the inside and their different functions. So there's a lot going on here, but a lot of these terms we've already covered in the presentation. So we've covered the vascular cylinder, which is where you're going to have your xylem and phloem located, carrying out their roles. We've already talked about the epidermal layers, the outermost layers. The only new layers that we haven't discussed yet are the spongy mesophyll and the palisade layer. So the palisade layer is this top layer, which is going to absorb light so that photosynthesis can take place. So even if I didn't tell you it was the top layer, if you know the function of the palisade layer in absorbing light, you know that light comes from above the plant more often than coming from below. And so you would expect the palisade layer to be located on top. So one way that I help students to remember this is that there's a city in Southern California called Pacific Palisades, and a lot of wealthy individuals live there. And so I think of them as getting the most light because they have one of the best locations where they live. And so that was just a trick I learned as an undergrad to memorize the palisade layer as being the top layer. Again, use whatever mnemonics help you, but I try to give you what I can in the hopes that it'll stick with you when it comes to an exam. Next, we have this spongy mesophyll layer. So this is going to be underneath the plant. So it's not going to actually get direct sunlight, but again, leaves do participate in photosynthesis, so it must have some other photosynthetic function. And in this case, it's going to be the region of cells that allow for the exchange of carbon dioxide. And if you'll recall, carbon dioxide is one of the necessary reactants that has to be present in a leaf in order to, or in a plant in general, to carry out photosynthesis. And it's this spongy mesophyll layer that's going to allow for carbon fixation, or the entering of carbon into the plant. Okay, so this is still our privet leaf. We've just zoomed in further detail so we can break some stuff down in more detail. This top region here is our palisade layer. And if you see all these dark purple structures that have been stained in our palisade layer, those are actually going to be your chloroplasts, which makes sense, right? Because that's the organelle that allows for photosynthesis to occur. So it makes sense that there should be a large abundance of chloroplasts present in the palisade layer. We also have our epidermal layer. It kind of looks like teeth, human teeth. So that might help you to remember it. And then we have our vascular cylinder. Again, this is where the xylem and phloem cells are going to be located. Again, photosynthesis is producing sugar and water that need to be transported, and the xylem and phloem are going to help carry that out. Okay, so this is still that same privet leaf, as you can see down here, but this time we're looking at the spongy mesophyll layer. And as we said, spongy mesophyll layer is what's going to allow for carbon to enter into the cells. And it's going to also allow for the exchange of other gases, like oxygen, right? That's where we get our oxygen, from plants. And because plants don't need it, they're going to expel it. So how that gas regulation takes place is through this small opening in plants called a stoma. So kind of like in the human body, we have our alveoli cells in our lungs that allow for the exchange of oxygen and carbon dioxide. And we're going to do the exact opposite of plants, right? So humans will burp out carbon dioxide, or they'll inhale oxygen. And plants do the exact opposite. They're going to inhale carbon dioxide, and they're going to, for lack of a better term, burp out the oxygen, right? So that we can take it in for ourselves. Now, this has to be carefully regulated, just like in the human body. And the cells that are going to do that are what we call guard cells. So that should be easy to remember because they're guarding the entryway that's going to allow for gases to come in and out. And this small entryway is called the stoma. So in next week's lab, we're going to actually learn about the hormone that regulates the opening and closing of the stoma. But if you can picture it, these guard cells here, when they shrivel and become super tiny, that's going to increase the stoma opening. That's going to regulate the ability for gases to more easily come in and out of the plant leaf. Whereas when these guard cells swell and become bigger, they're going to create a smaller opening or a complete closure of the stoma opening so that gases cannot enter and exit as easily. So here we have a different leaf. So the last leaf was a privet leaf. This is a zea leaf. And then, like before, this is a cross section. So here we're just looking at some terms we're already familiar with. We've talked about the epidermis, the vascular cylinder. This is the central part of the leaf. And then this region over here, we're going to jump into in further detail in the next slide. So in that highlighted region from the previous slide, we're looking at all these little circular regions. These are your vascular cylinders. And notice how, in comparison to our previous slide, we're seeing a lot of different images here, or excuse me, a lot of different vascular cylinders. So this leaf must be super important in carrying out the function of the xylem and phloem and transporting materials. So I'm not a botanist, but my educational guess as to why there's more vascular cylinders than in our previous leaf is that perhaps this leaf participates in a lot more photosynthesis than our privet leaf. And as such, it's producing a lot more oxygen, a lot more sugar. And so we may need to crank out more vascular cylinder factories to pump this material throughout the remainder of the cell. So this must be a plant overall that has a huge demand for sugar production and just photosynthesis overall. Now, it's hard to see here, but this arrow shows the stoma opening. So again, just like the privet leaf, the zea leaf also has a stoma opening, as well as the epidermal layers. Okay, so the final image we're going to look at is from an allium root tip. And this is a longitudinal section, so just a long image. And this is the root of plants. So as I mentioned at the beginning, shoots are the parts above the soil that are usually going to carry out their functions with the sun, because they have the direct exposure to the sunlight, whereas the root is going to be underneath the soil. So any nutrients that it's acquiring are going to come from the soil that contains bacteria or nutrients such as nitrogenous compounds that can allow for the plant to stay alive. So starting off at the top here, we have this region of cell elongation. So this is where you're going to have your cells becoming elongated in order to contribute to the overall structure of the root tip. And that cell elongation is going to in part be formed by this region down here where cell division is taking place. So this is where your mitosis is being carried out. And at the very bottom of the plant, a new term that we have is the root cap. So the root cap is at the very bottom, and it's going to secrete this mucus-like material, like what comes out of your nose when you're sick, that makes it easy for the plant roots to move through the soil. So basically, it's going to, in forming this mucosal-like material, it can easily snake its way through the soil in order to communicate with soil microbes or microorganisms in order to get materials from them. They can also interact with extracellular compounds, such as nutrients or nitrogenous compounds, minerals, salts, etc., that enable the plant to take up these important compounds that aid in its sustained growth. So just as important as a leaf is in acquiring and producing nutrients, the root cap is just as important. So this is that same picture. We're just zoomed in a little bit more. So this is our cell elongation. This is where cell division is happening. And finally, this is our root cap. And I'm going to zoom in one more time to our final image where you can see a close-up of cell division. And so if you look carefully, you can see different cells here are different phases of mitosis. So for instance, where I'm highlighting the mouse cursor, we can actually see telophase and cytokinesis taking place where you're having the sister chromatids split apart to opposite poles of what are going to become two new genetically identical daughter cells. Okay, so that concludes the presentation on plant anatomy and physiology. So again, feel free to go through this video as many times as you need to in order to review all the terminology. And again, I recommend doing that. As you can imagine, this material is going to be on upcoming quizzes or exams, so you want to make sure that if any material is confusing for any reason, please feel free to reach out to your respective 107 lab TAs, and we'll be more than happy to take any of your questions.